This Week in Microbiology is brought to you by microbe.tv and is supported by the American Society for Microbiology. To learn more about microbes, visit microbe.tv and asm.org. This is TWiM, This Week in Microbiology, episode 329, recorded on February 20th, 2025. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michelle Swanson.
Hello. Nice to see you all, and I'm excited about these papers.
I can tell when you're in Ann Arbor, you have the dark wood paneling around you, the knotty pine, right?
Yeah, I'm at home. This is not my university office.
Right. No knotty pine in the university, that's for sure. Also joining us from Charleston, South Carolina, Michael Schmidt.
Well, hello, everyone.
Michael, last night it was pretty cold down there, right?
It's still pretty cold down here. I think we're getting the Arctic blast that is affecting the rest of the country. I talked to my brother in Chicago, and he reported it was 21 below one morning when he went to walk the dog.
Wow.
Needless to say, he didn't walk the dog.
Well, here in New York, it's -7 Celsius.
Ooh.
Also joining us from Puget Sound, Tacoma, Washington. Sorry. It's the University of Puget Sound. Tacoma, Washington. Mark Martin, welcome.
Hi, everyone.
Is the weather pleasant there?
Oh, my goodness. It's actually quite nice. We had perhaps 3 days of snow last week, bunch of rain, and it's about 21 degrees in my office. Not too bad.
You have the windows open?
That would be Celsius.
It would.
21 Celsius?
Well, I've heard attacks on Fahrenheitism, so I do my best to avoid it.
You know that every country in the world except the U.S. uses metric measurements, and we, for some reason, can't get it together? Oh, my gosh.
Yeah, I don't think that's on Musk's list.
No.
But
No.
we do have the Gulf of America.
Let's talk about microbiology.
Yes.
Please.
Today we do have microbiology as usual, and Michael is going to start us off with Louis Pasteur reminiscences.
An unusual snippet. So as our tagline offers, our mission on TWiM is to explore with you the unseen life on Earth. Well, today we're going to do just that through an exploration of an article that appeared in the science section of the New York Times on February 17th by their longtime science correspondent, Carl Zimmer, who, by the way, has another fascinating article in this week's Times. Trillions of viruses live in your body. AI is trying to find them, that hopefully Vincent will talk about on TWiM. And that was published yesterday on the 19th. So the title of today's article is Louis Pasteur's Relentless Hunt for Germs Floating in the Air. Today, we're going to go take you back 165 years to the year 1860, where Louis Pasteur is going to be with us on our modern exploration of the unseen life on Earth.
Hey, can I ask you, Michael, you like this word germs?
No.
Good.
I haven't used it.
Mark, what do you think?
I talk about this. I've given lectures on this subject. And this is all about spontaneous generation. The term germ is viewed universally as negative, but no one thinks that about wheat germ.
Right.
That's
And
right.
germ is okay.
And so
True.
all Pasteur meant was a piece of life.
Yes.
And it's come to mean something else to us.
Well, he knew the term microbe. Why didn't he use microbe?
Don't know.
Right? He said he once said so in his era, a virus was any infectious disease causing agent. He said every virus is a microbe, which is right.
Right.
So he knew the word microbe. And Carl Zimmer, well, I guess most people respond to germs better
They
than
do.
microbes, right?
Yeah, and they respond this way generally, which is not.
They
not
generally
good.
are negative because we're not doing video, Mark. So your thumbs down sign is a thumbs down sign.
Michelle, do you like the word germs?
I mean, now that we understand all the benefits of our microbiome, I think it's misleading.
Yeah. It has a negative connotation, right?
Yeah.
Yeah. Okay.
Well, today as you might imagine, we're going to do a TWiM twist on how you go about refuting the theory of spontaneous generation using the methodology of today. And the reason for that is science is under attack, candidly. And so I think it's a service to the general public that authors like Carl Zimmer actually publishes such pieces in the popular press. So let's consider the world of 1860 that Pasteur found himself in, exploring along with one of his competitors, Felix Pouchot, who had challenged Pasteur's hypothesis that life came from life. And Pouchot offered his own interpretation that a vat of sour beet juice that Pasteur's student had asked him to investigate was a consequence of spontaneously generated microbes that were responsible for the transformation of the vat of beet juice, not into the desired end product, ethanol, but rather into something foul and ill-tasting, most likely vinegar. Louis Pasteur's experiments, particularly those that Carl describes so beautifully in his article, in the locations like the Mer de Glace, which is a glacier almost at the summit of Mont Blanc, where Pasteur and his trusty mule carried baskets of sealed long S-shaped flasks, or the swan neck flask, with a clear nutrient broth. Pasteur and his mule ascended up the mountain in order to test the following hypothesis: that the burden of microbes was higher in cities than in the clear, fresh air of the mountains, with the side bonus that they would ultimately serve to be pivotal in dismantling the long-held theory of spontaneous generation. Now, the theory of spontaneous generation had been around for many years. It was proposed that living organisms could arise from non-living matter. And this observation had persisted for centuries. You may have heard of the ether, where life came from. And Pasteur's meticulous experimentation and rigorous methodology provided compelling evidence against it. Now enter the French Academy of Science, where in their attempts to get to the truth of the matter as to whether or not there was such a thing as spontaneous generation, or whether Pasteur was correct that life came from life, they organized, as scientific societies might do back then, a competition to address the life from life or life from nothing hypotheses. Here, and I love Felix Archimedes Pouchot's middle name, and the not yet famous Louis Pasteur competed for a prize of 2,500 francs, which at the time was the equivalent of 727 grams of gold, or in today's dollar to gold conversion rate is about $69,000. So we're not talking pocket change, even in today's world. First, you need to consider where Felix was coming from. So let's consider his hypothesis that life came from nothing, or spontaneous generation. Where did he get the empiric evidence to make this observation? Well, for a long time, people observed that maggots appeared on decaying meat, or that microorganisms in broth left exposed to air would suddenly become cloudy, teeming with microbial life. The prevailing explanation was that life spontaneously arose from these substances. Now, for goodness sake, who has not seen that red stuff that appears on the hem of your shower curtain, or at the grout line in your bathroom, or even in the toilet? The surprising fact that that pink mold in your shower is not a mold or a fungus. Today, we recognize that pink residue is actually a type of airborne bacterium called Serratus marcescens, a gram-negative rod in the family of bacteria known as the Yersineaceae, and that's even associated with subgingival biofilms on individual teeth. Now, the pigment produced by this microbe is responsible for the color and produces this reddish-orange tripyrrole called prodigiosin And it's the same pigment that can also lend a red glow and actually stain our teeth. But I digress.
Yes,
It
you
wouldn't
do.
be a
Let's get up
TWiM.
to back to the
It,
Alps.
it wouldn't be a TWiM without one of my digressions. So, nevertheless, during Pasteur's time as the human race's collective understanding advanced, the theory of spontaneous generation faced increased scrutiny. So, enter Pasteur in the debate with a strong conviction that life originated from pre-existing life. Now, Pasteur's experimental approach, what today we would offer, was very straightforward. His genius laid in the ability to design experiments that isolated and controlled experiments, the same principles we teach the sixth graders in middle school, allowing him to definitively test his hypothesis. His most famous experiments involved the use of these swan neck flasks, and these flasks had long curved necks that allowed air to enter but trapped airborne particles, including microbes, in the S curve. And anyone who has ever been stuck on the S curve of Lakeshore Drive in Chicago at rush hour can attest. Even bacteria would have gotten stuck in the S area of the swan neck flask. They would never have reached the promised land of the nutrient broth. So what Pasteur did is he boiled nutrient broth in these flasks, effectively, and here I'm going to use air quotes, sterilizing it. He just boiled it to kingdom come, and it was before we knew about spores. But he then sealed the flask, and then in various locations throughout Lille, France, and the Alps, and wherever he happened to test, if spontaneous generation held, the microbes that were in if the microbes were to appear spontaneously, even in the broth of a sealed flask, you would effectively see turbidity. And the brilliance in Pasteur is he opened the end of the flask. He just broke the tip of the flask where the tip was below the horizon, if you will, from the media, so that the bacteria really would need a pressure differential in order to enter the flask. So when he broke off the necks of the flask, exposing the broth, and then when he tipped the broth into the S curve, the microbes quickly grew. But if he didn't tip the liquid into the S curve, nothing grew. So this demonstrated that the microbes came from the air and not the broth itself. Now, the mer de glace experiments that Carl Zimmer describes were based on the premise, as Pasteur had hypothesized, that the concentration of airborne microbes could vary depending upon the environment. To address this, he conducted this spontaneous generation type proof in diverse locations, including high altitude environments like the Mer de Glace, a glacier in the French Alps. By taking his experiments to these high altitudes, he was assessing whether the vital force required for spontaneous generation was more or less present in different environments. And by sterilizing the flasks and bringing them up to higher altitudes, he could expose them to air, then seal them to see if the microbial growth occurred. Now, the results from the plateau of the glacier did indeed confirm that the density of microbes were less, in that only one of the 19 flasks he exposed at the Mer de Glace grew effectively, serving to reinforce his observations. Fewer microbes were found at higher altitudes, correlating with the lower concentration of airborne particles at that altitude, where they were breathing clean mountain air outside of the hustle and the bustle and the horse manure and mule manure and detritus of modern cities of the 1860s. And again, air of the mid 19th century was heavily microbial laden, and Lille, France was no different than all the other cities. Horse-drawn carts, carriages, peoples, animals, so the air was heavily microbial laden. Now, the competition with Pasteur, who was this prominent proponent of spontaneous generation, he conducted his own experiments, which he claimed supported his theory of spontaneous generation. But the debate between Pasteur and Pasteur became a heated scientific controversy, And Pouchet's experiments were often criticized for lacking the critical, rigorous controls that characterized Pasteur's work. Pasteur's meticulousness and the clarity of his results ultimately prevailed, leading to the widespread acceptance of his germ theory and the rejection of spontaneous generation. So, for our discussion here today, let's reimagine Louis Pasteur's mirror to glass experiments using today's methodologies, where we can now leverage advances in the last 160 years of microbiology, molecular biology, environmental science, sampling
Autoclaves.
techniques, autoclave.
Autoclaves.
Well, that's the first step, Michelle.
Yeah.
Autoclaving and advanced filtration. So that's what I would have done first. And instead of simple boiling, we employ the modern methods of high pressure autoclaves to ensure complete sterilization of nutrient broths and glassware. Why? Well, today we know about spores and their ability to resist the process of boiling. Remember, Pasteur predated all of the work that was conducted by the American Can Company on their work with preserving foods through canning. They learned about spores. And let's not forget HEPA filtration would be used to create a built environment with as few particulates as even possible. Even within laboratory spaces in portable field units, any and all of us who have ever attempted to do definitive PCR, or the polymerase chain reaction, know that DNA is everywhere, and you can literally PCR a signal out of almost anything. And so now we get to the molecular sterilization techniques, and we're fast forwarding past the 1940s to gamma irradiation, or more importantly, cold sterilization using methods like ethylene oxide for heat-sensitive materials. But all of this pales with real-time monitoring that we have going on, and of all places, things like the New York subway system, the Washington, D.C. subway system, where we are continuously evaluating the air for not only select agents, but we're probably asking the question, what does the microbial load look like in these environments? And so we have come a really long way. And I'm reminded of some of the things I did with the Copper Project, where we did airborne microbial sampling, where instead of relying solely on open flasks, we used specialized air samplers that collected air, a known volume of air, and the air passed on to sterile filters that were actually a form of liquid media. I used many of those where we used a defined volume of air, impacted the samples on these gelatinized discs. Then we just simply dropped the disc into a tube of broth, vortexed it, and then did a serial dilution in order to figure out the concentration of microbes in the known volume of air. These samplers then simply allowed us to provide a precise assessment of the microbial load at different altitudes, under different varying weather conditions, using different interventions like we were using with copper air handling. Now enter the brave new world of inexpensive DNA sequencing, molecular identification. And instead of observing microbial growth, today we'll use whole genome sequencing, or we could use 16S ribosomal RNA sequencing for bacteria and archaea, not IKEA. I don't know what I'm doing.
Let's go shopping.
Well, they may have a swan neck flask at IKEA.
Yeah,
you never
might.
know.
It might.
And the ITS sequencing for fungi to identify the specific types of microbe that are in our airborne sample. And, Mark, when you teach this to the non-major biology students, how do they react to some of these new techniques and the, if you will, the absurdity of the hypothesis that you could have such a thing as spontaneous generation?
Well, and this is something I've taught for many years, and the old history is easy to make fun of. The
Oh,
idea the idea that you take some soiled cloth and some little bit of grain and it magically turns into mice. You and I laugh at that. But even Francesco Redi was able to show that those maggots that you talked about on decaying meat were deposited on the muslin on the jar. And even that wasn't enough. And I use this as an opportunity to talk about paradigm shift. I mean, one thing that you didn't mention is the people who were big believers in spontaneous generation. There was a group of them called the Vitalists. And the Vitalists believed that there was something special about standard food or whatever to promote the formation of complex life. So, when you boiled the material and didn't let it be exposed to air, you removed all of that. And that is why Pasteur invented the gooseneck, with the idea that there would be air that could travel to the actual broth with the
I thought
dust.
it was a swan.
He called it a swan neck.
I'm so sorry. So it's time to talk about the gooseneck. So the swan neck. So in any event, the dust, which is the primary vector, those fomites, collect here at the bend. And then what Pasteur would do is kind of flush it up a little bit like that. The whole reason of doing this is to put a stop to the Vitalists. And, I let students make fun of some of the old ideas, and I think that's fine. And then I remark on them to consider what they believe to be true that might be considered not to be true in the future. And that's why designing experiments is so important.
What I find really interesting about this is that Pasteur did a public demonstration. He said he decided to put on a spectacle, and he got a amphitheater filled with Parisian elites, which no scientist could do today.
No.
I'm pretty sure, unless it was Bill Nye, who's not a scientist, right? Can you imagine? And he showed that he could filter the air and put it under a microscope, and there were living things there. But that's how he often worked, right, in public. And it said here that the lecture ended with a standing ovation. His hunt for floating germs elevated him to the highest ranks of French science. Oh my gosh.
And he got his position at the institute in Paris that is now named after him.
It's amazing.
Right.
You're doing all the, and he and isn't it true that he and Lysenko had public debates as well? Because Lysenko was another
Yes, naysayer.
proponent of spontaneous generations.
Very end of his life. I mean, Lysenko is more in the 1920s, but
Okay.
But there were many people that refused to believe that these little germs of life were making all of this happen.
Yeah.
And in fact, this is what caused the problem. Oh, where's my brain? The fellow with sepsis in sick beds in Prague.
Semmelweis. Ignaz.
When I talk about Semmelweis, one of the things that I have to tell people is that Semmelweis didn't have a paradigm for how the sickness was being transferred. It's just dirty instruments or unwashed hands. But by the time that Pasteur had worked this out, there was something that could be connected to it. It made Lister's job a lot easier.
And Semmelweis's contemporaries were people like Florence Nightingale and, of course, Oliver Wendell Holmes Sr., not the jurist who served on the Supreme Court. That's his son. It was the other guy who was on faculty at Harvard.
I was also impressed listening to this story and reading about it, that Louis Pasteur packed all these flasks with, the swan neck breakable flasks on a donkey and went by himself on this journey up to a glacier.
The glaciers.
It's not like he had a whole team with him. He did this on his own. Yeah.
I mean, it was during very early part of his career. He was a young, if you will, assistant professor at the University of Lille in France. And he had this idea and he tested his hypothesis.
Driven by his own curiosity.
Yeah.
Clearly. Yeah.
So I wanted a contemporary version. I had a colleague at Columbia who designed a pump which would draw air through a 0.2 micron filter, and he put it in a backpack, and he walked through different environments in New York City, like the subway, and then he would take the filters back to the lab and see what was stuck on them. So a modern way of looking for stuff.
I also thought of the quest of is there life on
On
Mars?
Mars.
And can we
Yes.
find any evidence of microbes? And there's a lot of work is going into that.
Vincent, if I'm not mistaken, that was the study that showed kind of these cyclical changes in New York. And in the summertime, you had more things associated with chloroplasts because there were green things growing. And in the wintertime, you had a lot of things associated with the back end of dogs, gently put. Yeah.
Yeah.
Well, that's why tuberculosis dropped in New York City, is because we transitioned from using horses and horse-drawn carts to using trucks to deliver things into the Manhattan. And consequently, the horse manure would dry out and tuberculosis would spread into the tenements. And that was one of the single most reasons that we were able to get TB under control in the tenements of New York.
No, maybe they reintroduced horses into Kansas, huh?
That could be.
It's interesting, Michelle, what you said about life in other worlds, because do you know what the Lagrangian points are between the Earth and the Moon? So they're areas where gravity balances between the sun, the Earth, and the Moon. So you get what are very clear little pockets of dust.
Where there's no gravity that's pulling the particles one way or the other.
And there's not very much of it. Some people argue about it, but I think it's pretty clear from the astronomers that there's small amounts of dust in those Lagrangian points, L3 and L4 anyway. And I would love to see them take, I have a sample of it here. Do you know what aerogels are? Aerogels are I would like to run an aerogel through and then bring it back to Earth to find out if there are spores.
I know
or other evidence.
I would
of life.
know who would fund that, Mark. I'll give you his name later.
But I have to go myself. That's the problem. And I may not be able to come back. That's the problem.
Well, no, if you come back, nobody here would know you because they'd all be dead.
He wants to pack up his donkey with his aerogels.
Yes.
And head up.
But I mean, it's a really interesting question. And it could be none of that's true. Some people have done some work related to that. But I love this idea of how do you find out what's around you? And you'll notice in the essay, I mean, they expected not to see a bunch of stuff. But even then, Pasteur is saying maybe we brought it all with us, which, of course, is most likely true.
Yeah. Well, that's the problem with looking for microbes on planets. It's more likely you're going to bring it with you than it's going to be there, right?
Well, that's why people like Mimi Goldschmidt went to great pains to sterilize everything before they put anything into space. And I remember talking with her and Norm Pace about how do you know the spacecraft that you're putting into orbit is truly microbial free.
And how could it be, honestly?
It can't be. But you have to effectively engineer it so that when the sampler goes out, it's as clean as possibly it can be and actually have the control. That's the whole point of Pasteur's brilliance, is everything was controlled to the nth degree. And I think that's what we're going to have to do to look for life on distant planets, is to see whether or not we can actually detect anything novel, if you will.
Michael, thank you for that. And Mark, I have one word for you.
Yeah?
Ixotrophy.
Yeah. And I don't have a more clever name for it. I love talking about this with Elio. I had met Ralph Luhmann many years before. When it comes to bacterial predation, that is to say, bacteria that pursue and consume other bacteria, there are a couple of different routes we're familiar with. There are the ones, and in the paper I'm going to discuss, they actually, and now I'm going to speak sacre bleu, I'm not happy with this. They're calling Della Vibrio being something like a vampire. Well, let's not go there, because Della Vibrio, it lives within the periplasm of gram-negatives. Vampirococcus is something that, as the name suggests, and the Myxococcus that does the beautiful wolf packs where it basically secretes hydrolytic enzymes in front of itself. So Ralph Luhmann was the person many years ago who described certain close relatives to the gliding Bacteroides in seawater that appeared to be able to kill and consume other bacteria. Now, what's interesting here is not much work was done with that. Elio Schechter wrote a little bit about that in Small Things Considered many years ago. I have a dead frozen stock from Ralph Lewin's lab, as a matter of fact, of the organism he studied called Saprospira. So I was delighted beyond words to see a conglomeration of two different things coming together. The first was a series of posts on Blue Sky from Christoph, who's one of the co-editors for Small Things Considered, describing the paper that I'm going to talk about briefly. And I'll put a link to that in the show notes, because what I like about this series of, and I don't know what they call it on Blue Sky, I'm just going to say tweets. Anyway, the point is they often have small videos put into them, and it's actually put together by one of the authors. So it's really nicely done. In addition, there was a preprint of the paper, and it's come out in Science. So let me talk about that with you. The title of the paper is Mechanism of Bacterial Predation via Ixotrophy. Now, ixotrophy is a very fancy word, and I want you to all think flypaper. And because that's what we're seeming to see, except it's more complicated than that. What I like about this paper in the journal Science, and it came out in October of this year, author Lian, Amendola, Kansu Lee, Bartao, Chu, Furusawa, good old Martin Poltz, who I think the world of, Roman Stocker, Weiss, and Martin Pillhofer, who is the corresponding author. These are all folks that you're going to find pretty much in Switzerland and one person in Malaysia, which I find really, really cool. What I recommend everyone do is, in that same issue of Science, is a editorial on this topic by Simon Colton and Andrew Lovering. Andrew is a fantastic studier, and it's a really nice summary and has a beautiful set of images involved with it. So what I really like in the Science paper, far more in the preprint, is what you can see. Additionally, they have a beautiful research article summary. And what you're talking about here is this organism, which is Ariospira, and the fact it's using Vibrio campbelli, which is a common marine organism, as prey. And what is so interesting about this is they took a shoe, the shoe of scientific techniques, and they smacked the heck out of this system to understand it. And there are so many really good parts of this paper that are worth your time. So I recommend we get to it. Now, I am not looking at you when I'm doing this, so please shout at me if you have things to say. I love figure 1 here because it actually shows contact-dependent killing. And what has been very clear in the study of these types of organisms is that apparently the flagella of the prey are involved. And they have beautiful pictures of it. Turns out that Ariospira, which is a gliding bacterium, apparently has several cells inside kind of a proteinaceous packet. It's often called something like gauze. And the folks studying this did, I cannot tell you how many different types of techniques, ranging from looking under the microscope to knocking out genes to cryo-EM tomography. And it turns out that what appears to happen here with this contact-dependent and I love this fact that they're saying cell envelope-associated supramolecular machines. That is to say, large structures. And what appears to happen, and you can see this on figure 2, there are grappling hooks. Now, I will say when Christoph wrote about this for Small Things Considered, he was taken with the idea of bacterial piracy, and that was funny. But if you look on figure 2, it does in fact look like grappling hooks, and they appear to interact with the prey flagella.
Specifically, figure 2E, the cartoon, it really looks like a grappling hook that you have seen in the movies.
Well, the only difference is no one's swinging it and flinging it, right?
That's true.
That we know of.
That we know. There could be spontaneous generation. We don't know. There could be a little human in there.
It's really interesting to me because we don't understand how Ariospira is able to move. Gliding motility is one of those complicated topics. It's sometimes involved with pili, which is not how this particular organism moves. It's sometimes involved with essentially the nubbin of a flagellum, that is to say, just the motor, almost like imagine a tank. That's not what's taking place here. But there are some mysterious, and then there are the organisms that secrete exopolysaccharide at their distal end, commonly called snot rockets. There are others that create surfactants that decrease the surface tension in front of them, like their own slip and slide. All of this is absolutely true for some forms of gliding motility. That's not what this does. In addition to this, gliding motility on the part of the predator is not necessary to kill the prey organism. This happens both on surfaces and in liquid, and this doesn't always happen. I should say parenthetically, and I have more to say about this at the end that really excites me. I know, how could you tell? But the important thing is that this is an adaptation to low nutrient conditions. And that's the way predation generally seems to work. So this is a facultative organism, this Ariospira, that can live just fine on nutrient media, grow in liquid, grow on solids, but when it's hungry, it eats.
And when it's out in the fresh water, it's got to go hunting for its prey.
That's correct. And so if you imagine these grappling hooks, and I made a joke about them swinging around, but honestly, it's an interesting question because Vibrio campbelli just gets sopped up like crazy. And there are some movies associated with this paper that show exactly how quickly this goes down. And it turns out that what you're really looking at is related to a type 9 secretory system. And that appears to be what the grappling hooks are all about. Okay? So what we've done is we've grabbed Vibrio campbelli by the flagella and we pulled it close.
And they go to great lengths to demonstrate this is true. We were talking about Pasteur and his controlled experiments. They made a mutant that lacked flagella, and those Vibrio escaped the grappling hooks.
And it's important to remember the system that you study is important. There are lots of companies trying to develop genetic systems in relatively little known organisms. If they were using a different prey organism than Vibrio campbelli, they would have had trouble doing that particular experiment. But in this case, huzzah, you could make a knockout in the flagellum, and therefore you can see this to be the case. But what I like is that the actual killing is a type 6 secretory system, which some people describe as an inverted phage
tail,
which I can see why they say that, but I think it's older than that. So you have these two different secretory systems, one to grab onto the prey and the other to switchblade knife it, as I like to say.
Mm-hmm.
Mm-hmm.
And there are beautiful images of that. I'm looking at figure 3. And I mean, there's the cartoons, but what I think is so fabulous is the cryo-EM, because we just talked about cryo-EM on my own podcast 2 weeks ago.
That's a wonderful description of cryo-EM. If you haven't listened to Matters Microbial on the cryo-EM episode, you need to listen to it. It's fascinating.
So put that in the show notes. A link to that episode. Good.
Yeah, Ariana did such a fine job. It was amazing. So what I want to point out to people is that if you look clear the way out to figure 6, they're kind of showing you what's going on with the whole system. But there's one thing, and I'm really sorry if you're not interested in this, but I am. Turns out when folks were trying to knock out the type 6 secretory system in our Ariospira, they found out that sometimes it already was knocked out. And it turns out there's an insertion sequence, and it hops into the type 6 secretory scene. You can see this in figure 6. But here's the interesting part. You put that organism with the inactivated, and this is naturally inactivated, not by us, IS system into low nutrient medium. The IS pops out cleanly, and the type 6 secretory system is reactivated.
It almost looks like a cross between quorum sensing and phase variation.
Yeah. And a million years ago, when I was a young student, a friend of mine did work with what we called at the time Pseudomonas atlantica, which is now Pseudo-Ultramonas atlantica. and it makes an agarase. And it makes a lot of exopolysaccharide. And my friend found that this EPS had an IS element. The gene for EPS had an IS element that jumped into and out of it. And so when it was in the EPS gene, there was no EPS, so it was planktonic. If it hopped out, it made EPS and could adhere to surfaces as a dispersal mechanism.
And for our listeners who may not know what an IS element is, it's a sequence of DNA that generally has a transposase baked into the middle of it. And the transposase effectively cuts the ends of the insertion sequence. And again, this is a transacting factor, and it matters not where the IS elements may be within the chromosome, because the enzyme just is looking for sequence recognition. So you can get slicing and dicing all throughout the chromosome depending upon how many IS elements may be of similar nature and how Catholic the IS transposase is in sequence recognition.
And I'd love to share how I discovered this. I was able to get in touch with the first author, Yun-Wei Lan, and she described, she was a PhD student at the time. She described just a really frustrating series as she joined this lab and was trying to study this. And she had studied type 6 secretion systems previously. She couldn't get the mutant, or she couldn't get the bacteria to kill. And she thought maybe, it's something that I'm doing. Maybe I'm not culturing them right. But they looked by trying to purify this apparatus and then do transmission EM. And they found that the strain didn't have it. And when they did whole genome sequencing, born out of this frustration, there it was, an IS element, and not just one, right? They did this on a couple different isolates, and that's how they realized that it was a reversible event.
What I think is so interesting is, what is the signaling? We say low nutrient conditions and you pop the IS element out, or the IS element pops itself out, I realize. What is the signal? And I don't know.
Is it stochastic, Mark?
I don't know. And no one has really followed up on that thing with Pseudo-Ultramonas atlantica that I told you about. I always worry about something that's too good to be true in science that I want to believe. So I have to be very cautious about that. But this is a fascinating system. It makes so much
sense, because these really elegant structures, appendages, would be very expensive for the microbe to make. Right. They're really large. They can capture, it looks like dozens of Vibrio on one of them. So it makes sense that they only make it if they absolutely need it. So if there are plenty of nutrients around, we don't bother making this, going on our piracy mission with these grappling hooks and all. But if they're starving, they've got to go to it.
Speaking of taking the shoe of our techniques and hammering on a problem, they actually did stable isotope probing. Ooh. And they were able to show that the parts of Vibrio campbelli that get consumed end up in Aurea spira. Now that's not easy to do.
Really
but
elegant.
They really have a nice description for it. And I mean, this is not, last time I was on here, we were talking about some paper where they just really did that to death. And I like to see it. Although I understand that this level of intensity probably takes an awfully long time to put together.
And what impressed me about the grappling hooks that this microbe is making, I'm looking at it from the perspective of somebody who thinks about pathogens. And I'm reminded of cytophagia, which is another gliding bacterium that actually causes infections in the oral cavity. So I'm wondering if these grappling hooks are availing themselves to stick to our tissue and then consuming our cells.
Ooh.
Did you see the bit in there relating, and this is what Andrew Lovering wrote a little bit about. There's been some work recently about how Vibrio identifies hosts. And it's not anything simple. There apparently are these chimeric bundles of proteins that have been written about. I'd love to see This Week in Microbiology cover that in a way that probably my brain couldn't handle. I would get too excited. But Well, I would, because this is fabulous. It's a problem that's bedeviled me for many, many years. But what's interesting is they liken this kind of thing to what we're talking about with these grappling hooks.
Mm-hmm.
Even though I'm not sure about this, but it sounds as if this is, if not specific to Vibrio campbelli, almost pretty darn close is what I'm saying, which is not how Vibrio rolls.
No.
So, when you have any type of predator in a system, it will tend to diversify, after it clears out one. So this ability to spread itself to other things is fascinating. But I love the fact they actually did the single isotope labeling and were able to follow metabolites moving from the ex Vibrio campbelli into the growing Aureus spira.
And lest we think that it's one person like Louis Pasteur doing all of this themselves. What's really exciting about science nowadays is it's become much more common to do interdisciplinary work and reach out to people that've got expertise. And that was certainly the case here. So let me tell you that the first author, Yunwen Lin, studied plant pathology and microbiology as an undergraduate at National Taiwan University, and then went to a different institution in Taiwan and got a master's studying effector loading of type 6 secretion system in Agrobacterium tumefaciens. Was then inspired to continue and get a PhD and joined the labs of Pilhofer and Dr. Gregor Weiss, and there led the studies that we read about or heard about today from Mark. But she points out that this was a really valuable team to work with. They had a master's student, David Almondola, who did the painstaking work of purifying those grappling hooks and then doing some of the preliminary atomic modeling that led to the structure analysis. They had another collaboration with Kang-Soo Lee and Roman Stocker's lab, who did the Raman analysis that was able to show that constituents of the prey Vibrio actually made their way into the predator microbe. They also were able to collaborate with Nina Bartolo and Martin Polz at the University of Vienna, who did the metagenomic analysis, and in particular using samples from the ocean. And that some of their data suggested that there really might be some predation going on. So that was kind of the start of this project. They were also able to collaborate with single particle cryo-EM experts, which was essential because they tried very hard to use standard transmission EM, but they deduced that the staining wasn't sufficient to see them, so they had to go to the cryo-EM. So this is Zhu and Avasayev, collaborators that are authors on the paper. And then Go Furusawa is the person who originally isolated this golden spiral microbe that we heard about from a piece of seaweed in Malaysia. And that was back in 2014. So that's where they were able to get this microbe that they studied.
I would argue that predators are everywhere. We just have to look.
Yeah, they're in Washington, D.C., Mark.
She's also very grateful for the mentorship that she got. Her Gregor Weiss had initiated this project more than 10 years ago when he was a master's student in the Pilhofer lab, and is now a group leader in that lab and is studying this as well as some other cyanobacterium projects. She did say that this started out, it's a very exciting story, but it had its problems. We told you about the IS elements jumping in, and so the strains that she was working with kind of lost their magic, but they figured out it was a really cool explanation. And then again, the transmission EM was not sufficient for them to visualize these grappling hooks, and they had to go to the more sophisticated cryo-EM. So it shows that science, we call it research for a reason, not just search, but you get the right collaborators together and you come up with this really beautiful piece of work that is so interdisciplinary. And while we didn't go into it, they have quantitative data, well controlled. Everything is just so convincing and beautifully illustrated.
Mark, is predation always associated with grappling hooks?
No. So look at the myxobacteria. They basically secrete a wave of hydrolytic enzymes in front of them. So touching is not necessary for that. Same thing with Lysobacter, which is a cool name. Vibrio itself doesn't have a grappling hook. It does use type 4 pili to shoehorn itself between the outer and the inner membrane. And And these
Okay.
tripartite bundles apparently are how they recognize prey.
Now, why haven't the Vibrio made short flagella that can't be grappled but are still good at swimming?
I bet they do. And even though they had a figure where they talked about, I want to say 42 days worth of passaging,
Yeah.
you probably would be able to find similar
Yeah.
the short flagella. And I don't know off the top of my head about the structure of Vibrio campbelli's flagella. Sometimes they're very long,
Mm.
and there's some variation in how long they get to be, which would really be there.
Very cool.
You would become a hero to those bacteria by teaching them to make short flagella, Vincent.
Well, it's all they can.
Selective pressure will take care of it.
Yeah, so they already know how to do it, right?
It's called phase variation. They do it with an IS element, and they'd flip it out. They'd have the long flagellum for normal days, and then they have the short flagellum for when they encounter this
Yeah, that's
evil
good.
Oriella.
That's good.
Evil but beautiful. Remember that. Golden spiral.
It's like all Disney villains.
This
I think
grappling
I've been
hook
to that
just-
restaurant, the Golden Spiral. Huh.
This grappling hook is just amazing, isn't it?
Yeah.
That it predates human grappling hooks. And we think we invent things. I saw years ago there was a structure of a phage base plate solved with the spike, and the spike looks like a spike, like a knife that you would design to cut
Yes.
something, and molecularly it looks like that, and it used to puncture the bacteria, right? But again, all these design elements, they were figured out long before humans hit the planet.
Right. And this group did the painstaking work of deducing that it was assembled of multiple units of the same protein and go into a whole lot of detail that's really beautiful.
And I think it's important to emphasize that this is a predator that works on surfaces as well as in liquid itself. So, and how we don't know how it glides, that's interesting too. But that you see that a lot where people find out it's hard to figure out. So they just kind of like quit working on it because it's really hard.
They wave their hands,
Yeah.
throw them up in the air and say, I can't figure it out. Next student.
Mark, thank you so much for that
Sure.
lovely presentation.
Please, 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 if you're interested in this subject, the bit by that's the kind of the editorial on it it just takes you a little time to read, and it gives you a really good thumbnail sketch.
All right. It's TWiM 329. Show notes are at microbe.tv/TWiM. If you enjoy these programs, we'd love to have your support to continue them. If you want your nice dose of science, here's where you can come. Go to microbe.tv/contribute to help us out. And if you have questions or comments, TWiM at microbe.tv. Mark Martin, our TWiM adjacent Mark Martin, is at the University of Puget Sound in Tacoma, Washington. Thank you, Mark.
You're very welcome.
See you soon, I presume. Michelle Swanson's at the University of Michigan. Thank you, Michelle.
Thank you. Stay warm, everybody.
I saw a poll today on Instagram of the top wonderful places to live in the U.S. Do you know what the number one place was?
Well, I know in my heart it's Ann Arbor.
Yeah, it was Ann Arbor. I was amazed that
Wow.
Ann Arbor was up there. Look at
Yeah,
that.
Midwest is best, baby.
Well, it's until it turns red, Michelle.
It's
Then
nice
it's
and
not
blue
so-
now. We've got
then it's
3
not so
women in the
good.
top positions.
Yeah, but you guys voted anyway. Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone. Enjoyed it.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. See you next time on This Week in Microbiology.